Hello, in this video lesson, I'm going to have us do some practice with our Jamboard. And so if you could open up your mitosis practice, I'm using a copy, so it's a clean copy. And so this, I want you to get a feel for actually making a cell that's in these different stages and utilizing everything we know with our notes. So let's start with the interphase. Now the cell in interphase is not dividing. So we're going to show, here's my cell membrane and then the nuclear envelope. And so we're going to show all of our chromosomes all uncondensed. And so just drag your pile of chromosomes all, that was somewhat fun to draw. And I know it's kind of hard without having a scribe. So I went ahead and did it. So you can just grab your chromatin and just set it in the nucleus. And that's going to be our nucleus in interphase. And then we're going to add our centrioles. Now, a cell in interphase only has one set of centrioles. And so this cell is just doing what it normally does. And so I'm going to add a little sticky note, undergoing normal cell functions. And so feel free, you can stop the video and add your little sticky note. Notice the chromosomes are uncondensed. They're all just higgledy-piggledy. They're just all unfurled and are being used. And we'll talk about what that is in the next unit. So that's slide 1. Now, slide 2, let's make prophase. Now, I set up everything for us. And so what we're going to see, I made a nuclear envelope that is breaking apart. And we can make this a little bigger if that will help our room there. The other thing to notice is I gave you 2 centrioles. And I gave you 2 centrioles because they're going to be moving apart and they have duplicated. So after they've copied, they are going to start moving to the poles. And so I'm going to indicate that by drawing just a couple of arrows, showing that they're migrating to the 2 sides, to the 2 poles. And they would stop. Let's say this is North Pole, this is South Pole. They're going to stop when they get to the 2 sides. Now you can use the arrow shapes down here if you need to. There's our arrow shape. You can do that. And so a couple things to notice. Our chromosomes are suddenly nice and condensed. So compare the 2. Here is our interphase. All of the chromosomes are all kind of nice and thready, and they have gathered themselves up and pulled in. And let's count. Let's pause. Let's count upper chromosomes. You guys do this. How many chromosomes do we have? Pause the video. And you should have come together as a class. You should have 8 chromosomes. And we have, whoops, sorry about that. We have 2 purple, 2 red, 2 blue, and 2 orange. And remember, that's like having a set from mom and a set from dad. This is a diploid cell. And so we're going to add these into our nucleus. That is, starting, and we can have a little fun. We can move these around. Don't want to do that. We're going to move our chromosomes around just to show that they're not, they're not going to be perfectly, together or in the same orientation. So we should have 8 chromosomes. Now they're duplicated chromosomes, so that means we have 16 chromatids. Each chromosome has 2 chromatids. And they can overlap each other. That's no problem. That's kind of makes it a little bit more interesting looking. So there's our nuclear envelope breaking down. There's our prophase. All of the chromosomes nice and condensed. And then our centrioles that are moving. So do make sure yours is looking like this. You can hit pause if you need to catch up and make sure your prophase looks like this. Now, The other thing that I did forget, we need to add some spindle fibers. Now, you can go up here to your drawing and pick the pen, and I'm going to use blue because it shows up nicely. What we just want to do is just add some lines. They don't have to be perfect here. You just want to indicate that you know that these spindle fibers are being made. They're being made by the centrioles. So let's move on to our metaphase. That would be next. Now in metaphase, I just duplicated the slide, but let's get rid of the nuclear envelope. It's gone. So let's go up to the three little dots and you're going to click delete. We don't need that anymore. So in a cell with metaphase, the nuclear envelope is gone. Let's put our centrioles on opposite sides at the poles. And then what we want to do is we're going to put our little chromosomes. We can move our centrioles a little tiny bit because they'll line up really nicely. So we're going to move our chromosomes down the equator. Now they don't have to be any like specific order. We can have a purple one and then an orange one. It doesn't matter. Now, eventually, when we talk about genetics, we will learn that they need to be in a specific arrangement. But when a cell is dividing with mitosis, all we really want to make sure of is that the chromosomes are all lined up like little soldiers. And that means that when we go to separate them, they're going to separate nicely. And so there we go. They can overlap. There's no worries. Remember, the cell is three-dimensional, and we're just trying to do this on a very two-dimensional model. Now we've got one last thing to add, and those would be our spindle fibers. Now, again, it's going to be a little tougher. I've got a pen. You guys don't. So I'm going to use my pen, and you can do the best you can. And you just want to draw some lines. They don't have to be curved like mine, but you want to indicate that these spindle fibers are connecting to the chromosomes. And these spindle fibers, remember, they're microtubules. They're connecting to the center of the chromosome, and they're going to pull that chromosome apart. And so remember, the chromosomes look like X's, but as they get pulled apart, they yank, and then this part that's attached gets pulled, and they kind of collapse a little bit. That's okay. That's what they should do. So notice we've got all of our chromosomes stacked, and we are ready to separate them. So let's go to the next slide. And it starts looking a little ugly here, but let's make sense of this craziness. Let's put our centrioles, because they are at the opposite ends. They're at the poles. And let me show you what I mean by, so the chromosomes were together. So that would be kind of what the chromosomes were like in the previous slide, but they're going to then divide. So let me draw it for you real quickly. Let me use a highlighter. So imagine, if you will, you've got one copy and then a second copy. And as they get pulled apart, these copies separate like this. Now, because they're connected in the middle, as they get pulled apart, the whole thing kind of looks like two halves. You'll see with the GIF kind of why that happens. So let's make sense of this. So let's pull apart. We're going to pull apart our purple one, and you can hit pause at any point here. And then we're going to get an orange one. I think. Oh no, don't do that. So let me move my move this to the back here. So let's move my orange, and we're going to make two copies here. And so notice they're being pulled apart. And if we want to make sure I do it in the right order, red would be next. So let's put my red ones. They're being pulled apart. There we go. What came after red? Another orange. So let's put another orange. Let me scooch this up a bit. And we're going to make it nice and pretty looking with all the colors. And I got a blue and a purple. You guys can keep me honest here. So I got a blue, my other blue. And you'll notice I just made these a little smaller, partly because I needed more room. Where's my other purple? There we go. And move it down and move it down. All right, after the purple, I have a blue and a red. Blue. And you should remember we had 8 chromosomes to start. When they separate, these now get individually called chromosomes. So if we have done this correctly, each daughter cell should have 8 chromosomes. And we'll tally them up in just a second. And again, we're going to have our spindle fibers. And again, they don't have to be perfect. You just want to indicate they're getting pulled apart. Now, let's do a quick check. We have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes because they split. And then on the other side, 1, 2, 3, 4, 5, 6, 7, 8. And if we do this correctly, let me get the circles off. If we do it correctly, each daughter cell should have one purple chromosome there and then another purple chromosome. One orange, another orange. Remember, one from mom, one from dad. One red, another red. One blue, another blue. And each daughter cell should look like that. Now, the remainder would be telophase, and I'm going to skip to the next page. You're going to see a key for this. But here's telophase, and we can sort these out. So remember, we're going to have 2 purple chromosomes in each, 2 blue, one from mom, one from dad. And so we're just going to set these in here. And in telophase, remember in telophase, we're going to reform the nucleus. Now it's going to be tempting to say, oh, I'm going to put them together. But that is not, they are unduplicated chromosomes. Remember, you get one from mom, one from dad. And so they are distinct chromosomes. I think I had an extra set here. Let me get rid of my red ones there. And so, keep doing that. And so let me move these in. They should stay in the nucleus. Let's move that guy. And so there is our animal cell. Let's add a centriole outside the nucleus. And so again, if we do this correctly, there should be 8 chromosomes. Oops, I did something wrong here, guys. See if you can catch it. Hit pause and see if you can catch my error. All you be detectives. If you were very observant, you should notice that I goofed and I added an extra red chromosome over there. I should move that around. So if you caught that, good job. So 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. All right. We should have 2 yellow, 2 red, 2 blue, 2 purple. And that would be our animal cell. Now, in our plant cell, the only difference is we're going to add a cell plate. Now, I'm going to let you guys finish up yours. But let's add a cell plate down the middle. And that's how a plant cell is going to divide. So that should help you through your drawing. Go ahead and finish your plant cell, and then you can compare it to the key.